Today is my second presentation, that is photosynthesis, that is in higher plants. So photosynthesis, what exactly this process is and how this process takes place in higher plants. We all know that all animals, including human beings, that depends on plants for their food, directly or indirectly, right? So, have you ever wondered that how this plant produce their food, and how these green plants are much more efficient in producing their own food? So, what are their mechanisms, and how this mechanism is helpful for the production of their food as well as the production of oxygen? So this photosynthesis, as the name suggests, photosynthesis. Photo
What
means
is
the
going
light.
on? बैकग्राउंड में साउंड बहुत आ रहा है, क्या है? कोई है क्या पास में? कुछ लड़ाई झगड़ा कर रहे हैं?
नहीं सर,
आवाज आ रहा है, किसके साइड से आ रहा है आवाज?
वो बाजू वाले घर से।
बाजू वाले घर पे एक बार डांट के आओ।
खिड़की बंद करें।
चलो।
So ultimately, we know that photosynthesis is a process of through which the light is converted into chemical, that is a physico-chemical process by which plant prepare their food, right? So these are the outlines that we are going to discuss now. Today we will discuss only about the preparatory phase of the photosynthesis. So these are the outlines. What do we know about the photosynthesis, and where exactly this photosynthesis process takes place, and and what are the pigments that are involved in the process of photosynthesis, and which pigments are most efficient and responsible for the exact mechanism, and what are the reactions that include for this process, and during this light reaction process, which are the efficient product that we are going to use in the Kelvin cycle or C3 cycle. So these are some of the concepts that we are going to discuss now. So let's proceed. We know that this photosynthesis takes place in the leaf part, right? So we have already discussed the anatomical structure of the leaf. So we quickly recall this. This is the palisade mesophyll and the spongy mesophyll, which contain a large amount of chlorophyll. These palisades are the elongated cell which are arranged in a parallel fashion, and that too in a vertical manner. Whereas these spongy cells are small and elongated, and they are loosely arranged and contain large amount of chloroplast cells. So this is the enhanced structure of the chloroplast, well resolved structure, which is two membrane organelle. But this one is the outer and the inner, and here these are the grana, which are the stacks, which looks like a stacks of coin, and the single unit of grana is known as thylakoid. This thylakoid is the exact region where this light reactions takes place, right? So the one grana is connected with the another grana by the small lamella-like structure known as stromal lamellae, and the empty space which is filled with the jelly-like matrix known as stroma. Okay, so till here we already know that where the photosynthesis takes place, that is in the organelle known as chloroplast, and this plant absorbs light primarily using these pigments, which is present at the thylakoid membrane. So these pigments are embedded in the plant or in algae in a complex called antenna protein, and these proteins are responsible and these proteins work as a light harvesting complex, right? So this is the summary of today's lecture. So we know that the the reaction of photosynthesis is 6CO2 plus 6H2O, which gives C6H12O6 plus 6O2. So, so by visualizing this reaction, it shows that this oxygen has been evolved by the CO2, right? But by further experiments and all, by the reaction has been done by Von Neal and Robin Hill. They proved that this oxygen has been evolved, the molecular oxygen which has been evolved by the photolysis of water, not by the source of CO2, right? So the plant which produced their food in the form of glucose, this hydrogen and oxygen has been carried out by the which can be imported by the water, but the carbon source has been imported by the carbon dioxide. So by further reactions, by further reaction and further experiments, this is the exact mechanism and the reaction which has been proved that 6CO2 plus 12H2O, which gives C6H12O6 plus 6O2 plus 6H2O. For balancing the reaction, we have put this 6CO2 and 12H2O. So it gives, we should remember that this 12 molecule of water gives 6 molecule of O2, right? So in the light reaction, we know that this thylakoid membrane consists of 5 supramolecule complexes, which is more important, that we will discuss later. And this one most important factor that is associated with the PS2 system is the photolysis of water. That also we will discuss in a later slides. So how many pigments are involved in the photosynthesis? First of all, we will discuss what are the pigments and how they are involved and how they capture the lights, right? So photosynthesis have two type of pigments. One is the principal pigment or the main pigments, and the second one is the accessory pigments. So these are the supporter pigments. So this principal pigments is a chlorophyll A and the bacteriochlorophyll, whereas the accessory pigments are chlorophyll B, C, D, and carotenoid and phycobilins. These carotenoid are further classified are of two types, carotene and xanthophyll, and these phycobilins are phycoerythrin and phycocyanin. I have summarized this by showing this picture. You can see this chlorophyll A molecule. This chlorophyll, the molecular structure of this chlorophyll A consists of chlorine ring, and who has 4 nitrogen atom. You can see 4 nitrogen atom, and has several other attached sides chain and hydrocarbon tail. This is the hydrophilic pyrrole head and the hydrophobic phytol tail embedded in the thylakoid membrane, and the 4 pyrrole molecule linked together by the methane group. You can see this methane group to form porphyrin ring, and this maximum absorptions takes place at the red and the blue region of the light spectrum, and it reflects the green light. That is, you can see the formula is C55H72O5 and 4N Mg. Here is the structure of the chlorophyll A, and the only difference between chlorophyll A and chlorophyll B is the presence of aldehyde group at the side chain of the second carbon ring, and here it is a methane group. Now the second one is carotene. Its formula is C40H56, and maximum absorption takes place during the blue-violet region. Whereas this xanthophyll is also an accessory pigment in all plants, and it's the most abundant form of carotenoid is a lutein. You can see, and it is the structure of lutein is, its formula is C40H56 and O2, and the last one is phycobilins. These are they are also a light capturing agent found in the cyanobacteria, and this phycobilins This phycobilins fluoresce at the particular wavelength, and these are also used as a chemical tags in some research purpose. These pigments are capable of capturing the sunlight, and these are water soluble, and they are also the accessory pigments. And these phycobilins are of two types, phycoerythrin and phycocyanin. So these are also water soluble, and they are also found in cyanobacteria and red algae. So you can see the range, you can see the ranges, and at which this chlorophyll molecules or these other pigments absorbs the light. So you can see this chlorophyll A maximum absorbs the light of blue and red region, and same as in chlorophyll B. You can see the peak shows at the blue region and the red region of the spectrum. These are the another pigments that are phycoerythrin and phycocyanin, carotenoids, right? So as the leaf mature, the chlorophyll deteriorate, and these pigments are replaced by some of the carotenoid or xanthophyll. You can see the difference in the color, which represents the different pigments present in the leaf. So now we have discussed what are those pigments, and how they what are their function, and at which spectrum they absorb the light. Now we will discuss how they captured the light, right? So this is also known as a light reaction. We already know that this the light capturing agent are present in the thylakoid, and light reaction also takes place in the thylakoid membrane. So this is the thylakoid membrane, you can see, and it has 5 supramolecules, that is PS1, PS2, light harvesting complex, and CF, that is cytochrome B6 complex, and coupling factor. 5 supramolecules that we should remember. And this is the lumen of the thylakoid, and this is the stroma, chloroplast stoma. So first we will discuss what is light reaction and how exactly and efficiently this process. So before understanding and before proceeding with the light reaction, we should know the exact structure, right? So we know that the membrane of thylakoid consists PS1, PS2, cytochrome B6 complex, coupling factor, and light harvesting complex. So we know that these pigments, these are some of the pigments which are present at localized at the PS1 and PS2 system. But some of the pigments which are not a part of the PS2 or PS1 system, they are scattered at the region of lumen, at the region of membrane, and forms a light harvesting complex, which ultimately known as antenna, which captures the light. So we will discuss the efficient process between the PS1 and PS2. So first we will discuss PS1. So when the light falls on the PS1 system, so this pigments captures the light in the form of photons, and these pigments transfer those photons, those energy to the reaction center. And if we are talking about the photosynthesis in a higher plants, the reaction center should be the chlorophyll A molecule. Chlorophyll A. So these accessory pigments transfer their energy to the reaction center, and when this these reaction centers are full of energy now, so this emits their electron. So this electron first captured by the primary electron acceptor, and forms a hole inside this PS1 system. That is, the holes represent the devoid or the empty place of this electron. So this electron first captured by the primary electron and then transferred to the ferredoxin. This ferredoxin again transfer this energy rich compound to the plastoquinone, and this plastoquinone transfer this energy or electron rich compound to the cytochrome complex. This cytochrome complex use that energy to produce ATP in the presence of ATP synthase, and by combining this ADP with one inorganic phosphate, it forms ATP, that is the energy rich compound. And then the electron, which is now in a ground state, they will come again at their own reaction center and fill that holes, that position from where they are excited, right? So this was all about the PS1 system where this ATP has been produced. So during this process, we should focus on where this ATP synthesis takes place and how how they harvest the energy, that we will discuss later. This is about photosystem 2. So we know that here, when the light falls on the PS1 system, so simultaneously the light also falls on the PS2 system, right? So this PS2 system also forms, this PS2 system also capture the light energy. So here also there are some accessory pigments. These accessory pigments transfer their energy to the reaction center, and then this reaction center comes in an excited state and transfer this electron to the primary acceptor. This primary acceptor accepts this electron and gives that to the plastoquinone. This plastoquinone again transfer that to the cytochrome complex. So यहाँ पे जो PS2 सिस्टम है, इसमें हमें एक बात हमेशा ध्यान रखनी है कि जो PS2 सिस्टम है उसमें एक one of the most important mechanisms takes place, that is photolysis of water. जिसमें photolysis of water यहाँ पे जो हो रहा है, यहाँ पे हम easily देख सकते हैं that photolysis of water takes place in PS2 सिस्टम. और PS2 सिस्टम जो है वो 680 वेवलेंथ 680 नैनोमीटर का है, whereas दिस PS1 सिस्टम जो है वो 700 नैनोमीटर का है. सो ये एक फर्स्ट डिफरेंस है. सेकेंड डिफरेंस PS2 सिस्टम का ये है कि इट इन्वॉल्व्स इन द फोटोलिसिस ऑफ वाटर, जिसमें वाटर का ब्रेक डाउन होता है इनटू 2H plus and half O2 में. So if we add 2 molecule of O2, तो यहाँ पे 4H plus 4 इलेक्ट्रॉन एंड 2 ऑक्सीजन एटम होंगे जो अपने आप को रीअरेंज करके ऑक्सीजन बना देंगे, राइट? So we have already discussed how this process takes place and how these pigments capture the light. So plastoquinone and then cytochrome complex, then plastocyanin. So ये जो इलेक्ट्रॉन जो यहाँ से एक्साइट होके प्राइमरी इलेक्ट्रॉन एक्सेप्टर से मिलता है, एंड देन दिस प्राइमरी इलेक्ट्रॉन जो है वो ट्रांसफर करता है इलेक्ट्रॉन को टू द प्लास्टोक्यूनोन एंड द साइटोक्रोम B6 कॉम्प्लेक्स. यहाँ पे एक साइमल्टेनियस प्रोसेस होता है जो ADP प्लस इनऑर्गेनिक फॉस्फेट को मिला के ATP बनाता है, एंड देन उसका जो इलेक्ट्रॉन है जिसकी एनर्जी पूरे से हार्वेस्ट हो चुकी है, वो इलेक्ट्रॉन जो प्लास्टोक्विनोन के पास आ चुका है. अब जब when the light falls on the PS2 system, simultaneously the light falls on PS1 system. So यहाँ पे भी जो है वो एनर्जी रिच क्या बोलते हैं इलेक्ट्रॉन एक्साइट्स होंगे और यहाँ पे भी होल्स प्रेजेंट होंगे. सो जो होल्स यहाँ से एनर्जी जो इलेक्ट्रॉन के एक्साइट होने से प्रोड्यूस हुए हैं, वो इलेक्ट्रॉन्स ये वाले जो हैं वो इसको फिल कर देंगे, राइट? क्योंकि ये अब अपने उसी रिएक्शन सेंटर में वापस नहीं जा सकते, क्योंकि जो फोटोलिसिस ऑफ वाटर हुआ था जिससे 2 और 4 इलेक्ट्रॉन जो प्रोड्यूस हुए थे, वो इलेक्ट्रॉन इन द प्रेजेंस ऑफ मैग्नीशियम आयन इस होल्स को, यहाँ पे जो होल्स प्रोड्यूस हुए थे उसको फिल कर देंगे. सो जब ये इलेक्ट्रॉन जब अपना एनर्जी हार्वेस्ट करके जब वापस आएगा, तो उसके पास अपने ही रिएक्शन सेंटर में जाने के लिए कोई स्पेस प्रेजेंट नहीं होगी। सो ये जो इलेक्ट्रॉन है वो ऑक्यूपाई करेगा प्लेस 700, जो PS1 सिस्टम है उसमें जाके अपनी जगह ऑक्यूपाई कर लेगा, और ये जो इलेक्ट्रॉन है वो फर्दर कहाँ पे चला जाएगा, जो स्ट्रोमा में, जो यहाँ पे जो स्ट्रोमा है उसमें NADP जो को एंजाइम है वो प्रेजेंट है, उसका रिडक्शन होना जरूरी है। सो इस 2 इलेक्ट्रॉन को लेकर प्लस यहाँ पर जो 4 इलेक्ट्रॉन जो प्रोड्यूस हुए थे उसको लेके और 2H+ को लेके यहाँ पे वो अपने आप को इन द प्रेजेंस ऑफ NADP रिडक्टेज एंजाइम के प्रेजेंस में वो अपने आप को रिड्यूस कर देगा, किसमें, 2NADPH+H+ में। अब मेजर ऑब्जेक्टिव लाइट हार्वेस्टिंग कॉम्प्लेक्स का यही है कि जो प्राइम ऑब्जेक्टिव है हमारा फर्दर इज द प्रोडक्शन ऑफ एसिमिलेटरी पावर। सो यहाँ पर एसिमिलेटरी पावर कौन से हुए, ATP and NADPH. So the prime objective of light reaction is to produce asymmetry power, right? So यहाँ पे हमने asymmetry power बना दी है NADPH plus H plus and ATP, right? और ये जो है वो this photosystem to a non-cyclic photophosphorylation which always takes in place in a unidirectional form, or you can see the difference. Cyclic photophosphorylation involves only PS1, whereas this non-cyclic photophosphorylation involves PS1 and PS2 system, and the movement of electron is cyclic form, and the movement of electron is in a non-cyclic or in a unidirectional form. इसमें only ATP form होगा, right? You can see only ATP has been produced during the cyclic photophosphorylation. Photophosphorylation क्यों बोल रहे हैं? Cause in the presence of light, phosphorylation takes place. Photophosphorylation जहाँ कहीं भी होगा, we should understand that यहाँ पर ATP synthesis की बात हो रही है, right? So oxygen is not involved. One of the major drawback of this PS1 system is that कि इसमें जो है वो ऑक्सीजन का इवोल्यूशन नहीं, ऑक्सीजन का प्रोडक्शन नहीं होगा, और इसमें ऑक्सीजन का प्रोडक्शन होगा। Dominant in photosynthetic bacteria and dominant in green plants. Photolysis of water does not take place, and photolysis of waters occur. इसमें एक और एंजाइम है, इस जो सिस्टम PS2 के पास है, जो कि फोटोलिसिस वाटर का ब्रेकडाउन करने की, वो सिस्टम PS1 के पास नहीं है, और जो सिस्टम PS1 के पास है जो वो क्या है कि NADP, यू कैन सी PS1 के पास जो एक एंजाइम है NADP रिडक्टेज, जिसके प्रेजेंस में NADP जो है स्ट्रोमा में जो प्रेजेंट होता है वो H+ जो H+ कहाँ से इवॉल्व हुआ है, वाटर के फोटोलिसिस से, और इलेक्ट्रॉन जो यहाँ पे से आया था उसे लेकर अपने आप को रिड्यूस कर देगा NADPH plus H+। इसका रिडक्शन होना जरूरी है, कॉज हम at present we are in a preparatory phase, which means we are preparing some of the energy rich compound that we can use in a Calvin cycle or C3 cycle. So C3 cycle is itself is in the reduction phase, क्योंकि उसमें हमें फिक्सेशन करना है, रिडक्शन करना है कुछ कंपाउंड्स का, तो रिड्यूसिंग एजेंट हमारे पास होना जरूरी है जो कि ज्यादा अमाउंट में, राइट? तो उस चीज के लिए हम ये सारी फेजेस और प्रिपरेशन कर रहे हैं। सो बिफोर प्रोसीडिंग, यहाँ पर जो हमने बात की थी दैट द थाइलाकॉइड मेम्ब्रेन कंसिस्ट ऑफ 5 सुप्रामोलिक्यूल्स, अमंग देम वन इज द साइटोक्रोम B6F कॉम्प्लेक्स, जो कि B6F कॉम्प्लेक्स एक टाइप का एजेंट है जिसमें ATP सिंथेज नाम का एक एंजाइम है। इसी पे एक केमी ऑस्मोसिस करके थ्योरी है, वो डिस्कवर्ड हुई थी, जो हम अभी देख रहे थे। दैट chemiosmosis is a movement of ion across a semi-permeable membrane bound structure and down the electrochemical gradient. As an example of this would be the formation of adenosine triphosphate, by the movement of hydrogen ion across a membrane during the cellular photosynthesis, right? So अब आपको सिर्फ फोकस करना है इस पार्ट पे, साइटोक्रोम B6 साइटोक्रोम कॉम्प्लेक्स पे, राइट? सो साइटोक्रोम कॉम्प्लेक्स ये सारा का सारा जो है वो थायलाकॉइड के मेम्ब्रेन पे प्रेजेंट है, राइट? PS1 PS2 ATP सिंथेस दैट इज कॉम्प्लेक्स, साइटोक्रोम B एंड F कॉम्प्लेक्स, ये सारे के सारे जो है वो थायलाकॉइड मेम्ब्रेन के ऊपर प्रेजेंट है, और ये जो है वो ल्यूमेन का पार्ट है, और इसके बाहर में जो है वो स्ट्रोमा है, राइट? सो हमें एनर्जी कहाँ से मिलेगी? PS1 जो है, PS2 जो है, दैट वी ऑल नो दैट PS2 इन्वॉल्व इन अ मैकेनिज्म ऑफ फोटोलिसिस ऑफ वाटर, राइट? सो फोटोलिसिस ऑफ वाटर से जो मॉलिक्यूल जो H+ आयन जो रिलीज हो रहा था, यहाँ पे आप देख सकते हैं H+ रिलीज हो रहा है। सो वो कहाँ रिलीज हो रहा है? ल्यूमेन के अंदर, राइट? ल्यूमेन के अंदर PS2 सिस्टम, जो PS2 सिस्टम है उसमें से फोटोलिसिस ऑफ वाटर हो रहा है, और H+ जो है वो ल्यूमेन के अंदर रिलीज हो रहा है। Our main objective of our prime objective is to increase the concentration gradient inside the lumen, and और यहाँ पर concentration जो gradient है वो हमें in short gradient generate करना है। यहाँ पे H+ आयन का कंसेंट्रेशन बढ़ाना है, और यहाँ पे H+ आयन का कंसेंट्रेशन डिक्रीज करना है। सो वो हम कैसे करेंगे, और ये हम क्यों कर रहे हैं? Why we are increasing the H+ concentration inside the lumen to just to produce the ATP? अब ये कैसे करेंगे? सो H+ आयन का जो कंसेंट्रेशन है वो 3 रीजन से इसमें इंक्रीज होगा। PS2 सिस्टम में जो वाटर का फोटोलिसिस होता है वाटर का, उसमें H+ आयन रिलीज होता है, राइट? सो यहाँ पे 2 मॉलिक्यूल ऑफ H2O का जब ब्रेक डाउन होगा तो 4 H+ आयन हमें मिल जाएंगे। ये एक रीजन हुआ H+ आयन कंसेंट्रेशन को इंक्रीज करने का। सेकेंड रीजन क्या है कि जो प्लास्टोक्यूनॉन होता है, व्हेन द इलेक्ट्रॉन एक्साइट्स, और प्राइमरी इलेक्ट्रॉन एक्सेप्टर्स को एक्सेप्ट करते हैं और प्लास्टोक्यूनॉन को देते हैं, तो ये जो प्लास्टोक्यूनॉन है वो इलेक्ट्रॉन 2 इलेक्ट्रॉन प्लस 2 H+ आयन को फ्रॉम द स्ट्रोमा से लेके कहाँ पे डाल देता है? ल्यूमेन के अंदर, राइट? सो दिस प्लास्टोक्यूनॉन जो है वो ल्यूमेन में H+ आयन के कंसेंट्रेशन को बढ़ा रहा है, कहाँ से? फ्रॉम द स्ट्रोमा टू द ल्यूमेन। थर्ड क्या है? ये जो NADP है जो कि स्ट्रोमा में प्रेजेंट है, ये जो स्ट्रोमा में जो NADP प्रेजेंट है वो H+ जो है स्ट्रोमा में जो H+ आयन्स है उसको लेके अपने आप को PS सिस्टम 1 में जो NADP रिडक्टेज एंजाइम है उसके प्रेजेंस में अपने आप को रिड्यूस कर देता है NADPH+H+ में। अब ये जो H+ जो स्ट्रोमा में है वो कहीं और के नहीं है, वो बस यहाँ से जो ट्रांसफर हुए हुए हैं वो यही जो ल्यूमेन से जो स्ट्रोमा में आया है वही ही H+ आयन है। सो क्या होगा कि ये जो H+ आयन का कंसेंट्रेशन ल्यूमेन में इंक्रीज हो गया। अब ये जो H+ आयन है वो ADP प्लस इनऑर्गेनिक फॉस्फेट के प्रेजेंस में जो ATP सिंथेसिस है ये अपना चैनल ओपन कर देता है, और यहाँ से इसको बनने के लिए एनर्जी चाहिए होती है, और वो एनर्जी कहाँ से मिलती है? H+ आयन से। सो ये जो H+ आयन जब ल्यूमेन से स्ट्रोमा के तरफ आते हैं तो ये जो H+ आयन वो अपनी एनर्जी लूज कर देते हैं, और ATP सिंथेसिस जो है वो एनर्जी को ATP में कन्वर्ट कर देता है, राइट? सो दिस वाज ऑल अबाउट द concentration gradient of H+ and how this ATP has been produced, and how this concentration how the H plus ion concentration decreases in the stroma and increases in the lumen of the thylakoid. That's it. This was all about the preparatory phase of photosynthesis, and in next lecture we will discuss how these agent, this NADP, H plus and ATP are used in this in the Calvin cycle, and we will also discuss what are the modification or what are the means how the plant modified their mechanism to prevent the loss of oxygen, and how efficiently they produce their food.